Okay, we get to move on to the chemical senses, which if you've taken sensation and perception with me, you know that this is olfaction or smell is one of my favorite topics too, is it's just a fascinating. It's just another thing that really fascinates me, how this works with our memory systems and everything. I'm going to try and keep it short so that we finish it today so that when we are discussing e-reserve articles that you're at least have a little bit of background for getting into potentially that one about olfaction. Okay, I'm going to start this by talking about the crucial nature of olfaction. I sometimes, when I have a face-to-face class, I ask the question of if you were the almighty being and you created a one-celled organism, what sense would you give that organism to help it survive? And many people, even though here we are at olfaction, and I feel like I'm kind of giving them the answer, people say vision. Vision is really important to us. We are a very evolved species, but vision gives us that really the distance of information. But remember, we have to point our eyes at something for us to actually see that information. It's what's would be more helpful for a one-celled organism or back in the evolutionary chain would be olfaction. It would definitely be a chemical sense, and this particular chemical sense is one that means we don't have to actually touch or ingest whatever chemical that might be poisonous, right? So that we can sense that chemical without having to touch it or ingest it in any way. And here are some examples of how it helps other animals survive, and it helps us as well, and we're not as aware of much of this, and our survival doesn't depend on it to the same extent. But how much pheromones influence us could be something we question. But we find food and mates, or other animals really find their food and their mates based on their sense of smell. If you've ever had a dog in heat, you know that the male dogs can find that dog, or a cat in heat, it's really attracting those mates from far away, potentially. We respond, or other animals respond to predators. What is being shown in the picture there is a mouse that has lost those olfactory receptors for smelling or sensing the cat, and the cat has just had a big meal, so it's not playing with or trying to kill the mouse. But typically, mice, even brought up in a laboratory for generations where they've never experienced a cat, they're going to respond to the smell of a cat being in the room. They're going to go into their hideouts. It helps us to recognize dangers. It helps us to recognize poisonous foods. So in our world, the smell of gas and smoke can tell us there's a danger. Poisonous foods, if something's rotting, we can smell that as well. This is something that is that we've lost a great deal of because of the amount of change and that we've had because of evolution and because of our powerful visual abilities. But just as a comparative, a bloodhound has 220 million olfactory receptor neurons, so there's actual sensory receptors, compared to us we have about 20 million, and about 5% of their cortex is devoted to olfaction, where it's about 0.1% for us. And I know I very briefly talked about that idea of cortical magnification of the fovea, that we have more representation of this important information, but so other animals, more of their cortex is devoted to olfaction than to other senses. It's very important to them. And even humans can follow a scent trail to some extent. So what this is showing in the picture is a trail of, I think it was a chocolate oil, if I remember correctly. And you can see in the orange lines, so the sort of yellowish line is the chocolate oil, and in the orange lines, they had blindfolded these people, put gloves on them, and they were following the trail using their sense of smell. And you can see that she keeps coming back to the center line and is following it pretty well. And we get better at this with practice as we are using that part of our brain. Okay, and we have hundreds of different olfactory receptors, or olfactory receptor neurons, about 350 according to Linda Buck and her colleague Axel. Thousands, so different animals are going to have different numbers of receptors. Thousands in rats, those animals that for whom the olfactory sense is more important. They're going to have more than us, so they can smell a wider range of chemicals and smells. We might do this in comparison to the fact that we have 4 kinds of visual receptors, or rods, that are 3 kinds of cones. 350 gets us into a pretty complicated situation. They are alive for about a month. If you remember from the neuroanatomy chapter, or no, it was the development chapter of discussing how we do create new neurons later in life, and that the olfactory receptor neurons were one example. They also have a kind of metabotropic effect. So one thing you'll notice in the picture here is the strand of protein that is snaking across the membrane 7 times. I very briefly said that for vision in this class. Sometimes I talk about that a little bit more, but this is a similar kind of setup that we have for the visual sense, where that opsin in that case was snaking across the membrane 7 times. And when we have some chemical that this strand is going to respond to, it has a metabotropic effect on the neuron. So those slower effects that influence a G protein. And I've taken this picture from E. Bruce Goldstein's Sensation and Perception book because there are aspects of this picture that I like better. It is much more clearly a drawn figure than what is in your textbook. And the picture in your textbook is also very good and helpful. It's on page 218, and it shows really where this is as far as in relation to the brain. But what this shows with these colors, and of course we wouldn't really have these colors, but the point of him providing these colors is to distinguish between those different olfactory receptor neuron types, so that those different types of receptor neurons are going to be responding to different specific chemicals in the environment. And so if I take and remember there are 350 about in humans, and this is showing 3, and the 3 are either orange, they're green, or they're pinky purple. And obviously, again, they're not colored like that, but I'm just going to take the pink colored neurons. You can see that they're going up through the cribriform plate there, through this little bony structure, and that particular type of neuron is communicating to a particular glomerulus, or really they communicate to a pair of glomeruli in the olfactory bulb. So we're seeing the olfactory bulb at the top there. The other things that are being shown here are the pigment epithelium, so those support structure cells, as well as, so up where the little cilia, what look like little hairs that come down underneath the nose, are in the olfactory mucosa, as there's this moisture up there that allows for chemicals to be absorbed easily into these olfactory receptor neurons, or by these cilia, so that we can experience our sense of olfaction. Again, he's just showing us the types with those colors, but I think it's helpful. The olfactory bulb shows patterns of activation that reflect different chemicals, the chain length of those chemicals. So chemicals that are similar are going to have somewhat similar patterns of activation, and they're going to get slightly different as we get a different chain length. But the olfactory bulb also reflects different perceptions. So what's being shown, I'm going to start on the right in B, is something that is having the same smell, that you can see the plus limon or minus limon. I don't really know my chemicals very well, but whatever that means, that's the change in the chemical. But otherwise they're similar, and so they have pretty similar patterns of activation. They also smell the same. So again, slight differences because they are different chemicals, but pretty similar. Whereas if we move over to A, okay, the plus carvone, minus carvone, that is, again, the chemicals themselves are very similar. This is the only difference, is plus carvone or minus carvone. So they look relatively similar in their patterns of activation, but these two actually are perceived differently. They smell different from each other, and you can see what he's pointing to with the white arrows. What he's pointing to with the black arrows is the places where they are very much similar. With the white arrows, he's pointing to those areas that are different, so that are active, more active in the left, the plus carbone example, versus more active with the reds and yellow colors in the right, in the minus carbone example. So that is reflecting that these are smelling different, the different perceptions. And what this picture is showing in a very simplistic way is how we are getting that information, okay, from those olfactory receptor neurons in the olfactory mucosa to the olfactory bulb, and then that information is going directly to the piriform cortex, which is our primary receiving area of the cortex for olfaction, as well as to the amygdala, so already very clearly in one of these emotionally associated memory areas. Okay, and notice that for every sense that we've talked about so far, and so usually I've done taste and hearing as well, but all of that goes through the thalamus, and the thalamus is doing that, summarizing, organizing, filtering information based on top-down processing and bottom-up processing of the brainstem and so forth. But this is really going directly from that olfactory bulb, the very first place it synapses, coming from the receptor neurons themselves, into the piriform cortex and the amygdala. Piriform cortex communicates directly with this orbitofrontal cortex. So the orbitofrontal cortex is that area of the prefrontal cortex that is just above the orbits of the eyes. So really where we have some higher level processing. And here for the orbitofrontal cortex, we're putting together all of the information from smell and taste when we're experiencing the flavor of our food, smell, taste, hearing the sound of food, the looks of food, and the feel of food in our mouth. All of that comes together at the orbitofrontal cortex. In addition, just so I say this here, because this is really, as much as I'm preparing you for reading that very complicated article, if you do, on olfaction, that there are other regions that the piriform cortex is communicating with, including the entorhinal cortex and the pararhinal cortex, which we don't really talk about very much in this class, if we do at all. If we do, it'll be when we talk about memory. These are places that, areas of the cortex that communicate directly with the hippocampus. So really into these memory areas, emotionally associated memory areas. This explains why we have those experiences with smell where we can be brought back to very vividly to a certain time in our lives if we're smelling the smells of our grandmother's house or something that was in our playgrounds or our lives when we were very young, especially if we've moved away and we come back. It's very noticeable as we haven't experienced it since then, and it'll just bring us back because of how smell goes very directly into these memory areas. There are individual differences in olfaction that we don't see in the same way in other senses. There are some to some extent for taste, but to a large extent, right, as far as we know, our cones are behaving normally or similarly to other people. Our rods are responding similarly to other people's rods. Here, two people chosen at random most likely differ in around 30% of their olfactory receptor genes. So what exact olfactory receptor neurons and exactly what they are responding to were going to be different from each other, where some odorants are going to be experienced as stronger, some as weaker than each other, some as more pleasant, some as less pleasant, which really explains why we have these differences in our preferences for various smells. Some more individual differences. Again, these are just things that are pretty fascinating to me. One, we see a general decline with age, that neurogenesis slows down. We also see that we can kill off these olfactory receptor neurons just like we can kill off our taste cells with a chemical so quickly that we're not going to replace them. Or we can kill them off with smoking. It looks like smoking cigarettes, that this does damage to those different receptors and that we're not replacing them as quickly as we need to replace them all. We see a sharp decline, can be an early symptom of Alzheimer's and of Parkinson's, as we'll talk about when we talk about Parkinson's. We see in various research studies, women are more sensitive, have a more sensitive sense of smell than men. This is in all the cultures that have been tested and at various ages. We also see that young adult women are more sensitive to a faint odor that they are repeatedly exposed to. And this is true for young adult women, not true for men, not true for prepubertal girls, not true for women after menopause. I have the definition here of pheromones. So chemicals released by an animal that affect the behavior of others of the same species. And that is what your author has in there, but I'm going to go ahead and say that these chemicals released by an animal can affect the behavior of animals of other species, as I'm sure you've experienced, or if you have a dog, if you've had dogs, that if somebody is around who's afraid of dogs, that your dog knows that by the pheromones that are being released by that human. He discusses a little bit here the vomeronasal organ. So in most animals, this is an organ that is sensing these pheromones and so communicating to the brain about mates and predators and danger and ovulatory cycles and all menstrual cycles and all of that about other animals. Again, if you know how if you've had a female dog, you know that they can attract those male dogs from pretty far away when they are in heat. Same with the female cat, you might have experienced. For us in humans, that vomeronasal organ is a vestigial organ in humans. It's tiny and it's not responding to pheromones, but we do still process pheromones. We have receptors among those olfactory receptor neurons that are responding to pheromones. One study that examines this is from Kathleen Stern and Martha McClintock, which is often referred to as the menstrual synchrony experiment, although I'll go ahead and say that this menstrual synchrony experiment was originally conducted in 1971. Other labs tried to replicate with some changes in the definition of exactly when they were going to count the people as having the same menstrual cycle, but Stern and McClintock replicated in 98 with a different measure, as I'm going to talk about here. But again, other people have not replicated, other labs have not replicated. But for any female who's lived with another female, you know that our menstrual cycles do synchronize, and it looks like this is happening via pheromones. So in their study, what they did was they took underarm secretions from donor women who were in a particular phase of their menstrual cycle. And then they treated that so that there was no smell to it with alcohol. And they rubbed it under the noses of recipient women and told those recipient women not to wash their face for, I think it was 6 hours. So they did this so the donor women were originally in their late follicular phase of the menstrual cycle. And what this did was accelerate a surge of luteinizing hormone, which then shortened the cycle of the recipient women. But they did this at another time. So with the donor women, they later took underarm secretions during the ovulation phase, so a later time of the menstrual cycle. And then the recipient women, we see a delay in that surge of luteinizing hormone, which lengthened their menstrual cycle. So it looks like this is the reasoning for why our menstrual cycles synchronize when we live together. Some other examples, since those haven't been replicated, it's always good to have a few more examples. Some research where they have taken women's sweat, okay, where they made the women sweat in shirts, and again, they treated those shirts so that we're not smelling, so it's not about the smell or olfaction, and especially if those women were in the ovulatory phase of their menstrual cycle, this would increase a man's testosterone. Okay, so in their participants, their male participants, we see an increase in their sex hormone. They've done this flipped around, different research group. take the sweat of males as they are working out in their t-shirts, treat those t-shirts, and what they found was an increase in cortisol in women. So we've increased the woman's stress hormone. Okay, I sometimes start the importance of olfaction with one of my little factoids from John J. Rady's A User's Guide to the Brain. It's a really fun book if you're at all into this stuff. He talks a little bit more for lay people than a lot of people, but he's still using a lot of the neuroanatomy as well. But a lot of these fun factoids that I have marked in my book. So prior to mating, the female silkworm releases 0.01 millionth of a gram of the chemical bombacol, and that is enough to attract 1 billion males up to 3 kilometers away. And I often make the joke when I'm starting all this that it might be a little bit scary to be a female silkworm if I'm attracting that many males. But they only have 5 days to mate and to reproduce, so it makes sense that they want to be able to attract them from far away. But if you've seen your female dogs or female cats in heat and the way that they can attract these males from pretty far away, it might be a little bit stressful and scary to be a female in heat and to smell males, whereas our smell clearly increases their sex hormone. And I think that's kind of a funny and interesting bunch of facts. Okay, so there's some information about pheromones. It looks like I'm going to stop here today because getting into taste, I'm just going to be able to do maybe a few minutes, and that seems silly to start and do a few minutes, like 3 or 4 minutes, and then finish on Monday anyway. So I will try to shorten taste a little bit and finish up with taste so we can move on into movement. We do want to start using this information to move around eventually. And then so my summary slide is basically just coming back to these pictures that I feel like are very informative of how we are taking in those chemicals from the air. They are dissolving and affecting, depending on the chemicals, affecting different olfactory receptor neurons, which are then communicating to the olfactory bulb. So different types of olfactory receptor neurons are influencing different glomeruli, which is how we got that, what was called a chemotopic map. I don't think I used the terminology, but that mapping that you saw with the higher activity in the olfactory bulb is really telling us about the chemical. It's telling us about the specific chemical, the chain length, and really pieces and parts of that are the differences in the perception of the smell as well. And then the picture that brings us into the brain, so that we're going straight from that olfactory bulb into the piriform cortex and the amygdala, and not going through the thalamus, very important, since that is doing some of the summarizing, filtering, organizing information. And really, if you think about how the amygdala is helping us with our classical conditioning, that's pretty important. And then we're going right into the orbitofrontal cortex and into various memory and emotionally associated memory areas. Okay, so we will finish with this on Monday, and we'll have the discussion about those journal articles that are relating to sensation and perception on Friday.